at some of the activities that we have planned or have been involved in, and listen to two very exciting and interesting talks today, and then a bit of Q&A questions, and then finishing it, wrapping it all up. So for people who are new and are just joining us, we wanted to discuss maybe a little bit as to who we are and what our main goals are. So we're a network, a community of researchers and practitioners, which are working to create an equitable, user-led, decentralized, and sustainable diagnostic innovations ecosystem in the global health. Really, the goal of this network is to enhance access to R&D for global health and diagnostics. We have a couple of objectives. We would like to share and transfer technological knowledge into the makerspace domain, sharing innovations and good practices amongst ourselves to help our research, and democratize access to point-of-care diagnostic development. We're hoping to accomplish this by doing, just as this meeting, online and in-person workshops, develop frameworks that can be shared across the network for developing or establishing new initiatives and new labs and new projects, and to create and distribute and make available a set of toolkits and resources and SOPs that could be shared. So wanted to give you a bit of a recap from our last meeting. Our first meeting had about 19 countries, participants from 19 countries, about 57 participants. And for this particular one, we had registered 172 participants. However, I can see only 42, so maybe they will trickle in as we go along. And we've increased the number of countries representative to 27. So in terms of people who are aware of the network, we can see a significant increase. We hope to be able to involve people as we go along a little bit more. I know we've been a little bit quiet on the Slack platform as we're getting that organized and set. So please keep an eye on that. It should be getting a lot more active fairly soon. Kind of a bit of a recap. If you want a bit more details, you can take a look at the Nature Microbiology. We had a post there. So here are the three kind of points that I wanted to remind everyone and maybe bring them to our attention, is that as I mentioned before, the creation of the network has been motivated by the gap of the health needs and the capacities to develop solutions in the global south. That we want to focus or look into frugal diagnostics, which are about the development of simple diagnostics by the people who need them. Then we had two exciting talks, one by Dr. Otinieto, who described transformations that we have seen across the continent due to, research in malaria and HIV, but he also pointed out that there's still numerous needs that are continue to exist. And then he also pointed out that currently there isn't too much intentionality about looking at the local context, and perhaps that is one of the things that we could be aware of as we're developing new options and we're developing this new network. Our second talk was by Dr. Zibusido, who highlighted that we see a fairly low adoption of new technologies, and we're presuming that that is due to the limited involvement of an end users. And perhaps as we are developing our network, this is another thing to consider. How do we forge better and more effective communications with the end users to help the adoptions of new technologies that we are developing? We also participated into two events. One was the Global Health Workshop in August, the 1st and the 2nd of August, where we had Harry and Maiwen, who chaired a panel discussion on sustainability, and also had given the keynote talk on sustainability and equity in the global health diagnostic space. And then we also had a second blog published in Nature, I think, or at least is submitted and might have not made it out yet. We also had Maywin giving a talk on the Reclone community meeting about open microfluidics for diagnostic, which was greatly received and reviewed, and loads of people became really interested. So I'm sure some of them have joined us perhaps today as well. And I think at this point, I will pass it on to Harry for introduction of our speakers.
All right, thank you very much. And it's great to introduce our speakers for today, who will be presenting 15 minutes in total each. And the first speaker is Professor Duedu. And Duedu is an associate professor of molecular biology and biotechnology at the Department of Biomedical Sciences, University of Biomass. He completed a postdoc with the African Partnership for Chronic Disease Research, University of Cambridge. And he held the Novartis Global Health Fellowship at Research in Cambridge, MA, United States. And another postdoctoral fellowship with the Tackling Infectious Disease to Benefit Africa TBA program with the University of Edinburgh. He is also a medical laboratory scientist and a fellow of the Higher Education Academy UK. In response to the COVID-19 pandemic, he led a team to establish the University of Health and Allied Sciences COVID-19 Center to test for infections and later on to undertake research using genomic sequencing and other technologies to support the public health response to the pandemic. We will now hear his talk titled Community Mobilization for Leapfrog Diagnostic and Research Capabilities During COVID-19 Pandemic. Over to you, Dr. Duedu.
Thank you very much. Can I get to share my screen?
Yes.
Okay.
Yes, just 1 second. I think you are able to share your screen right now.
All right. Can you see my screen now?
Yes, I can, and I'm hoping that everybody else can as well.
Yes. All good for me.
Okay. Let me pull this aside and then. All right. So good evening, everyone, and happy to be here today to share this brief talk with you. One of the things I will aim at doing is to just share the broad highlight of our journey, how we came about setting up the US COVID-19 Testing Center and some of the things that we have done. I'll aim not to make it too much of the science, but to highlight the efforts that we put in place at a point in time where there was a critical need for the diagnosis. So as Harry has mentioned, I work with the University of Health and Allied Sciences. This is our School of Basic and Biomedical Sciences campus in Hull. And this is where we actually set up the center to be able to do our activities. So my talk today will go through this brief outline, which I would go through. So on 28th of March, February 2020, that was when Nigeria had reported their first case of COVID-19. The Vice Chancellor, Professor John Japong, called a meeting, bringing together the whole teaching hospital and the university to strategize for a response. And this, one of the key reasons was that when you look at Ghana and Nigeria, whatever happens in the other country most likely will get to the other. So it was really important for us to do that. So I called one of my PhD students, John, at the time, and then I told him that there was a need for us to get ready and prepare the lab to be able to do this activity. And so John and I went to the University of Ghana, Noguchi Memorial Institute for Medical Research, where we went to look at the system they were using for the diagnosis in order to learn from them and then also set up our own activities. From there were a series of activities that we did towards our preparation to start with the SARS-CoV-2 diagnosis. So there was a community briefing where we brought people together to tell them exactly what we were hoping to do in terms of testing and our response to the pandemic. So I had my colleague, Dr. Bachiri Adomu, who is a laboratory physician, coordinating between the lab and then the clinic activities as well. So we went on to do the resource and team mobilization. And this was where we needed to be quite strategic and innovative in our activities. So we looked around. I mean, at the time, we didn't have the setup ready to do the testing that was critically needed. And so we did the initial mobilization where we brought together resources from research projects that were ongoing in the lab. We had some donations from seeding labs, from Novartis, and then one other interesting thing that we had to fall on is to put our hands in our pockets and then get some money to get some basic things that were not readily available. Then we did propose a team of scientists across the university, which were assembled and commissioned by the Vice Chancellor to be able to get started. And we're able to do all of this between the ending of February, and then we were able to start testing on 27th April 2020. So this is one of our teaching labs, which we managed to commandeer to start with the testing. So we cordoned it off. So it will be solely for receiving samples and then preparing them for the testing activities to be done. Another key element which I think we were able to do, which is unusual, is that we didn't have the resources. The government didn't give us the things that we needed to work with. And so we decided to fall on corporate Ghana. And so we had support from the Hoteliers Association, for example, within the Volta region, coming on board to make donations of personal protective equipment. We had them making donations of a fridge, freezer, and others. And then we had other entities within the country and in the region also coming to give us some other donations. Some of the banks came to give cash donations, which helped us to buy additional PPEs and some of the consumables which we needed. And so we realized that at that point in time, the community mobilization, because of the need that was there, people came on board and were able to join hands, not just the science, but also to put the structures in place to provide the necessary testing. So these were the earlier activities where we had the hotels and other entities coming to donate from Veronica buckets where we could wash our hands, soaps, and all of that to be able to make the work done successfully. So we also at a point had the National Trust Fund supporting us with a pickup truck, which became very useful because we didn't have an incinerator on site. And so our waste management, we had to carry the waste from the lab premises to the teaching hospital where there was an incinerator to be able to do that. And with the stigma and all that, it was really important that we had a dedicated vehicle to be able to do this particular activity. So initially, when we started at my lab, we had the X16 well qPCR machine, Chai single channel, which we started with. And then along the line, the International Atomic Energy Agency And the Ghana Atomic Energy Commission decided to do some donation to support the testing activity. So they were able to get us an additional PCR machine and some other equipment that were very useful in terms of we expanding our capacity to do the testing that we needed. Another challenge which I believe most other African countries face is stable power supply. And at this point, at the peak of the pandemic, we were facing that same challenge. And so we lobbied some organizations around, even including parliamentarians, and we were fortunate to get the Volta River Authority to buy a standby 250 kVA generator to support the lab. And so this ensured that at every point in time, when the power went off, we had power to be able to continue with the testing activities. As I mentioned earlier, one area that we needed to build capacity to was a team. And so at the time, the university was closing a lot of its activities. We made an invitation to faculty and staff across the university who had some background in clinical diagnosis and molecular biology. We brought them together. We designed and delivered some hands-on training so that everybody will come up to speed with the techniques that we were going to use for the testing. I should mention that because of the expense associated with doing molecular biology in our environment, not many people do basic things like PCR and all of that. So this was a really important step for us to be able to expose everyone to doing RNA extraction and doing some basic PCRs so we can bring the testing that was critically needed at the time. So beyond that, what are some of the things that we're able to do? We were able to deliver routine testing and surveillance for SARS-CoV-2. We're also able to deliver some genomic epidemiology monitoring activities, and then to introduce some research, particularly on differential diagnosis, which I'll mention shortly. So in terms of the testing, we did the routine testing. We tested for travelers. We did the surveillance as well. So for people who came into the country, tested positive at the airport, samples were sent to us to be able to test and do sequencing as well for the PCR. We did our activities covering the Volta and Oti regions. So these are 2 out of the 16 administrative regions of Ghana. And in addition, we also covered for the Sojamani district of the Eastern region and occasionally for the Adan district of the Greater Accra region. These were all areas which were closer to us, and so it made sense bringing samples to our labs other than going all the way to Accra. This, I would skip it because the figures have updated. So the other thing is genomic epidemiology. When there were concerns over variant circulation and evolution, we needed to piggyback on our existing sequencing capabilities from existing projects. So we had some projects ongoing looking at the microbiome as well as genomics of antimicrobial resistance. And so we mobilized the existing resources to be able to do the sequencing of SARS-CoV-2 genomes. We tried as much as possible to, what we did was to select the samples which came from all the regions. So in this map on the left, you can see the country divided into the 16 administrative regions. The areas we covered are the areas marked in the blue, the red, and then part of the green, which is the Eastern region. So we had samples coming from all of these areas for us to do the testing for. So some of the key things that we're able to do was to be able to contribute to highlighting which variants characterize the first, the second, the third, and the fourth waves. And then we also were able to help contribute to identifying the fact that there were many variants that characterized the pandemic in our regions. And as time went on, there was always a transition of a major takeover by new variants along the line. And we did also contribute some of the data to some Pan-African efforts as well as national efforts to be able to get a better picture of the pandemic in Africa as well as in Ghana. Let me skip this. So the other thing that we did was the differential diagnosis work. And we realized that a number of the samples which we received were suspected for SARS-CoV-2 by test, and then they were negative for SARS-CoV-2. And so we began to question what other pathogens could be there. So that led us to start a project on looking at other bacteria and viral pathogens that were present within the sample. And we did this together with the Kumasi Center for Collaborative Research, where we shared samples between the two sites. This is actually an ongoing study, which we are still going back to the samples to be able to get a better picture of what was there. But so far, some of the things we've realized were that some of the PEEP samples which came in suspected of COVID, some were due to influenza A, some were due to TB, some were due to strep pneumo, and then we had some also due to respiratory syncytial virus. A good number of these cases were not co-infections. Co-infections were there, but they were very few, which means that in such situations where most of the efforts tend towards the agent, which it's the prime suspect for the pandemic, may not be the key. And it's important that we don't lose track of other potential pathogens so that in the heat, we don't lose them and miss the diagnosis and their management. We also thought that it was important for us to develop diagnosis internally. And so we also undertook some activities to develop some isothermal kits. A lot of these activities are still in development, where we are currently optimizing them, and we hope to share some of the data in the near future about some of the things that we did. We also did some collaboration with the West Africa Center for Cell Biology of Infectious Pathogens, looking at the therapidemiology within the country as well. Beyond the activities that we did, we realized that by building those capabilities, we could offer training and capacity building. And we thought the testing should go beyond the centralized testing that we were doing at the heat of the pandemic. And so we had support from the WHO and the Ghana Health Service to organize training for laboratory personnel within the Volta and OT regions. And through this training, we're able to build their capacity, and currently they are able to do a lot of the testing on site in the health facilities, shortening the time between when the test is needed and then when the results are ready as well. Now, I want to acknowledge my team, Dr. Aibo Jani, Dr. Jones Jenfi, and Dr. Grace Pelly, who helped me a lot to coordinate all the activities that we did. And then also to the different organizations that provided funding and in-kind support in one way or the other to make our activities successful. Thank you very much.
Thank you very much. It's really a great Till Bachmann as well, who will be sharing with us a 15-minute presentation. But before that, I'll just briefly introduce him. Professor Till Bachmann is Chair of Molecular Diagnostics and Infection at the Center of Inflammation Research at the University of Edinburgh. Till is the EMR strategy lead at the Edinburgh Infectious Diseases, Chair of the Edinburgh AMR Forum, and co-director of the University of Edinburgh's Fan Fellowship Host Institution Program. Professor Bachmann is expert in point-of-care detection of infectious diseases and antimicrobial resistance, conducting research at the interface of biomarkers and rapid diagnostics, including the coordination of the UK-India project DOSA, Diagnostics for One Health and User-Driven Solutions for AMR, as well as a range of other international networks. Till fulfills several industrial and institutional advisory roles worldwide, including chair of the Scientific Advisory Board of the Joint Programming Initiative on Antimicrobial Resistance, judge for the Longitude Prize on Biotics, advisor for CABEX, and founding member of the BEAM Alliance Diagnostics Task Force. We're going to hear his talk titled Co-Design Diagnostic Solutions to Tackle Antimicrobial Resistance in the Community in India. Thank you very much, and let's hear from Till.
Thank you so much, Harry. Can you hear me? Very good.
Loud and
Okay,
clear.
so yeah, thank you so much for the invitation and opportunity to speak. And as it was said, I'm talking about a very specific case of diagnostic development, which we are embarking on specifically in India and specifically on antimicrobial resistance. As it was said, so I have a few activities which I'm engaged with, and many of them are on antimicrobial resistance and diagnostics. And I want to point out that we are working also with the Fleming Fund, and we are a host institution for the Fleming Fund Fellowship Program. And I recently took over as a co-director for this host institution program. So we're working in Africa, in Kenya, Malawi, and Uganda, and entering our next round of activities there. And as you may know, Fleming Fund is targeting the capacity building around antimicrobial resistance with on lab capacity, but also with the One Health objective. Now, I also want to point out that we started some years ago, and this is you see here in the I hope you see my pointer in the middle of the slide on the left. There an activity, Antimicrobial Resistance Diagnostics Challenge Competition, AMRDXC, and that is something which I would like to also highlight later, maybe in the discussion, because that's something I can see this network being specifically suitable for in terms of collaboration and taking those ideas forward in terms of seeding innovation in country with respect to the respective innovator base. Now, let me start with I don't need to introduce to you, I'm sure the reason why we're interested in antimicrobial resistance, the global health challenge. Diagnostics is an intervention which has been identified long ago as one which can be very impactful, and it has also been featured in numerous global health initiatives. And you may be aware that just recently the WHO, the World Health Organization, has launched their global research agenda for antimicrobial resistance in human health. And if I put here the link, so you can look it up in case you haven't seen this yet. There is a nice recording of the webinar as well. So I was presenting the diagnostics of bacterial infections, and this is a good introduction to today's event. So diagnostics of bacterial infection, what we have is that very often if a patient comes from the community into a healthcare center, we have the situation that therapy has to start immediately, but current diagnostic takes too long. And that's why we're developing rapid diagnostics, and many of us here in the call and beyond that. The issue with this situation is that we don't have these rapid diagnostic tools, we don't have enough implementation, but the bigger issue is that many of the global population, and in fact 47% of the global population, do not have access to diagnostics at all. And that is the problem. So how do you get diagnostics to all of the population globally? How do you reach people in the community with your diagnostics or with the healthcare impact which you want to achieve? Now, the WHO has presented a range of priority subjects, and one is that as it described here, is to investigate and evaluate rapid point of care diagnostics, and as you can read here, and what they really want to achieve here is a test which tells if an antibiotic treatment is needed and if the treatment works, and it should be available anywhere. So a very simple test, possible to do everywhere. And when I presented this, I thought, oh, this is actually exactly what we are doing with DOSA. And DOSA, our project, which I will describe to you in a second, is doing exactly that. A simple test which can be developed anywhere, and is then giving information about if a treatment is needed. Now, the DOSA project is outlined here. So we were running this in the first phase, so UK-India collaboration. with settings in 4 settings in the UK and a number in India. And we have 3 domains where we're looking at in the human, in the dairy and aquaculture settings. And we took a different approach to what usually people do in diagnostics. So we did not start with the technology development and then looked for application. A worst case scenario. This is what you should never do. You should always think about your end user and your end user application. But here we really took it to a next level. We went out and did design ethnography in these settings, in the 3 settings. We did baseline assessments in terms of what is the antimicrobial resistance to, situation there, what is the user doing in these situations, who are the stakeholders there, in a way to then come up with suggestions for development of target product profiles for diagnostics in collaboration with the diagnostic technology developers, which then develop prototypes, which then are tested in the respective settings and then further evaluated and or further progressed and evaluated. Now, I want to describe to you the situation we had with urinary tract infections in community care in Assam. So we are active in rural Assam and peri-urban situation and very rural situations, and looking into primary healthcare centers. And the initial idea was, oh yes, we collect urine samples there, we look for the antimicrobial resistance, and then we see what the user needs are there for diagnostics. Now the situation was that we actually didn't receive many urinary tract infection samples because, well, as we learned, people didn't come or do not seek medical care if they suffer from urinary tract infections. And of course, then if we would come up with a diagnostic solution in primary healthcare centers, we wouldn't succeed in tackling antimicrobial resistance or urinary tract infections. So what we thought is that we actually need to take a different approach. And one was also driven by the fact that we had identified through our collaborators that the awareness of antimicrobial resistance, the awareness of urinary tract infections was relatively low. And that, of course, is combined with the barriers to reach the healthcare facilities is combining a difficult situation. Now, so we went on and tackled this, but I wanted to show you that we had in the project, we have numerous technology solutions which we are all capable of and wanted to develop. So for example, on top left corner here this is an electrochemical sensor which is easily detecting antimicrobial resistance genes at point of care, just connected to a laptop, so we can easily make molecular diagnostics combined with microfluidics, etc. So that is something which we can deliver, but we found that this is not suitable for our application here. So we had a lot of community engagement. We worked with the community healthcare workers, the ASHAs in India or in Assam there in our setting. We had co-design sessions where we gather their input into the design of a diagnostic. We then had workshops in Edinburgh where colleagues from India came over, and we had further progression of the designs of diagnostic solutions. And we quickly realized that this is not just the diagnostic technology, but it's the bundle. It is the everything around it. It's the healthcare information, it's the way we deliver it, it's the way the data are used, it's the place where the diagnostics is applied, etc. And this was developed, as I said, and then also tested in real-life walkthroughs in Assam with community members in order to get further feedback and further progress the design of the solution. And this is what we came up with. It's very simple, and it's exactly as it's depicted here. It is a test which sits in basically a lot of healthcare information and a lot of advice. And the crucial aspect of this is that we provided pictorials, guidance on the urinary tract infection symptoms, as you can see here. So these are the pictorials which point to the origin of the symptom. And this was, for example, a good way of vocalizing symptoms, starting to speak about symptoms and start help to identify that actually urinary tract infections are present. In fact, there's also, of course, a language issue. Urinary tract infection is not a term which wasn't used. There was a local term for the condition, which everybody had to learn, vice versa. And with that, we came up with this solution. The diagnostic test itself is a very simple dipstick, and you may be aware of the urine dipstick, which is used routinely in diagnostic laboratories. We developed this further so it can withstand the environmental conditions. And is also biodegradable as we dispose this. And of course, we also looked into data capture, but I don't have time to look further into this. So the strip itself is very simply manufactured by screen printing on paper-based substrates. It's very simple, we kept it simple in terms of its chemistry, its dry chemistry, which we're applying here, so we can manufacture them in relatively large quantities. You may ask, why are these guys not using a commercial urine dipstick? They are available all over the place and can be cheaply manufactured. In fact, they are not so cheap. They are, most importantly, not environmentally stable, and they are quite, there's an issue of biodegradability. So if you can think about using such a test in the community and then disposing this test, you're adding to the plastic waste. I haven't outlined this, but the idea is that actually the test is done by the community member in the community with the guidance of the community healthcare worker, the ASHA, and is then, if positive, leading to the patient then coming to the primary healthcare center and being fast-tracked there and provided antibiotic treatment or the standard healthcare. Now, we tested the UTI test strips. We validated them. We validated them in Edinburgh in the medical school against the standard urine dipstick test and standard UTI diagnostics, so with culture and microscopy, and we did the same in the Silchar Medical College, which means in India, in Assam. So, and the test performed very well, and this means that we could then move forward into the next phase. Now, what we learned from this is that it's really important that you develop your diagnostic technology, but you, of course, need to think about your stakeholders and the network, the health system you're operating in. And what we found is that without the test information, the patients were pretty much lost on their own. But with the and the ASHAs and every player in the situation, but with the test, there is a lot of opportunity to structure the relationships and the connections between the respective players, from the community all the way up to the hospitals in terms of antibiotic provision, healthcare provision, and also the data capture. What was really important for us was the fact that as we operate via the ASHA network, so the community healthcare network, we amplified a lot the reach of the diagnostic, and we also, of course, got interest from outside of the network or outside of our project of people having a lot of interest in our approach and wanting to get access. Once you start verbalizing and depicting a situation, is very common, urinary tract infection, people realize that something can be done about it, and then obviously a lot of interest was generated. Now, I'm just back from a trip in field work in India, where we're now testing these tests in the communities under the conditions. It was monsoon season, so very high humidity, very high temperature, and testing the actual performance of the strips themselves over time, so stability test, performance test, before we then go further for rollout in the community as we are planning it. I don't have much more time to go in detail, but I wanted to share one more example where we did this also through the DOSA project in a very similar fashion. Again, having a very simple test designed together with aquaculture, with shrimp farmers, in order to detect antibiotic residues. Before we started this project, we identified people, the farmers in particular, had very challenging connections and interactions with the respective stakeholders and authorities with regards to providing samples and getting the results back farmers are very interested not to use antibiotics, obviously, because that gives them the possibility to export and achieve higher revenues for their product. And with the diagnostic, there's an opportunity to take control of that situation and make sure that you are informed and make an informed decision. The test itself is very simple and can be deployed for farm use. And with that, I want to acknowledge everyone who funded us, who collaborated with us, and specifically the partners in country, and you for your attention. And I'm very happy to answer any questions. Thank you very much.
Thank you very much, Prof. Bachmann. I think this is really a great presentation, and this takes us to the next round of the events, which is the Q&A session. And I would start off that by engaging the entire event. And if you do have questions, please feel free to ask in the chats, and we're going to highlight those questions in the Q&A session. But I do have some questions I'm going to use to curate these sessions. And the very first question is open both to Professor Duedu and Professor Till as well. In your work, especially on how to engage, build, and test for developing diagnostics, there are, of course, some challenges that you have faced. It would be good to listen to what some of those challenges are with regards to frugal diagnostic developments and how you were just generally trying to maintain accuracy and reliability in developing those. Any of you could take these questions, this first question. Maybe we can have Till go first since he just finished, and then we have Kwabena.
Okay, so the question was about challenges and the challenges in developing diagnostics. Now, this is what my research is mainly focusing on. So we have traditionally people develop diagnostics and face technical challenges, but then once they have developed the diagnostics, there's an issue with uptake and use. And so the barriers for development, uptake, and use are the biggest issues for diagnostics. Now, this is a big discussion to be had around this. The traditional challenges in terms of technology are, as I outlined just before, in terms of stability of the assay. Very often we produce assays and then or tests, and then when you put them out, they are not stable. And this is one example of what is really a challenge, and where the real world very quickly kills something which you have developed in your test. In terms of antimicrobial resistance, there is another one which is actually which is really challenging. It is not just one pathogen or one in a disease or one type of infection. You have multiple pathogens, you have multiple drugs you can use, you have multiple infection types, and that makes the thing with antimicrobial resistance so challenging. So in short, that's one issue. In terms of adoption of a test, there are again numerous challenges which one has to face there. The biggest, of course, is first of all, you need to show validation. You need to show that your test is performing as a gold standard test. In our area of antimicrobial resistance, we often face the situation that there is no real good gold standard test. So if you want to be faster in point of care, then of course this fast and point of care is not always similar as the gold standard, which is laboratory based and, for example, growth based in our case. Then I'm just continuing because there are so many. Yes. I think there are many other areas. I would mention just one more, and that's around regulatory aspects. If you develop a test and you want to roll it out, obviously for application, you need to have regulatory approval from your respective country where you want to use it. That becomes a challenge if your market in the country is small, and you are, so if the domain for the regulatory authority is small, and that small because the country is relatively small, and so the market is small. So the money you need, the budget, the resources you need for development of a diagnostic then become less. It becomes less attractive if the market is small. So regulatory aspects are very are a big issue, and this is something where, for example, harmonization strategies can be used to address this. And then just the last point, and this is for diagnostic developers, this upfront investment in the development, finding investors for that is very challenging because the markets are not so attractive. And finding situations and solutions where there is a guaranteed buyer, like a government, that helps a lot. And this is addressed in the discussions around antimicrobial resistance globally and various situations where we find incentive models for diagnostic development. And a lot is learned, and this is where I'm handing over to Kwabena, a lot is learned from COVID. In COVID, for COVID diagnostic development, there was a guaranteed buyer in many cases. And so diagnostic developers could develop their technologies, and then there were governments picking things up and buying the tests from them, which helped a lot to facilitate many of these developments. I'm stopping here. I could go on.
Okay.
But I'm stopping here.
Probably we hear from Kwabena, and probably we have some specific questions for you too, and then we come back to that. Over to you, Prof.
All right, thank you very much. That I would come from the user perspective as well, because it's also very important for diagnostics. So with a unique environment where we find ourselves, so for example, in Ghana, where there is resource limitation, usually one of the challenges which everyone talks about is funding. But beyond funding, there are some other challenges that we do face when it comes to the diagnostic. One of the key things is accessibility. So there is this kind of huge bottleneck when it comes to, even when you have funding, getting access to the necessary diagnostics that you want. And when you want to develop diagnostics, for example, developing PCRs and other simple molecular diagnostic that you can use when there is a need, you don't easily get the resources that you need. And so that is one of the major gaps which needs to be filled, particularly in resource-limited settings. The second element has to do with optimization. So during the pandemic, we realized that, so if you listen to my talk, one of the things I mentioned was that from the onset, the resources were not coming, and so we had to find resources. But towards the end, when donations had come in and all of that, now there were a lot of resources that the government had procured. And a lot of these kits came in and they had very short shelf lives. And so a lot of the tests had actually expired. But then one of the things that we realized was that we could actually repurpose and re-optimize them to do other things. So for example, a lot of the differential diagnosis which we are doing, we realize that when you take the kits which had the RT master mix separate from the primers and the probes, you could actually re-optimize the RT master mix and all that, and then you could actually use it for both RT-PCR and even traditional PCR. So other than throwing away the enzymes, you're able to use that. We realized that you could also make use of the extraction kits and others in a similar way, where you do the optimization to look at where the gaps are to be able to fill in. And this is one major challenge that many other labs do face, where they have a lot of consumables which will expire. Sometimes during procurement, you want to get a lot so that when you need again, you don't suffer, but then they expire on you. And I think these are some of the things that could actually be done in terms of that particular challenge. And then the other thing I would talk about is the capacity building. So a lot of the times we need to be able to build capacity and encourage scientists, particularly in resource-limited settings, to be able to go beyond just following the protocols, to explore, tear apart the protocols, and then build it again. So building the confidence, building the capacity so that we can tailor things to our own needs. I think these are some of the challenges and the way that I think we can resolve some of them. Thank you.
All right, thanks very much. So before we move on to the next round of questions, in the interest of time, I would employ us to keep the responses a bit brief. So to you, Prof. Duedu, there's a question about what other technological, be it assay equipment and other needs you may need that would be helpful to allow you to access collaborations from the global north to support the kind of work you're doing, especially when you were speaking to inaccessibility. I think from Ayo, he wants to know what are some of the technological needs that would help you to overcome some of this access barrier.
So for us, some of the technological needs, one had to do with the computation, the bioinformatics, in terms of when it came to LAMP assay development, for example, we realized that designing the primers for LAMP, which can be quite tedious if you're able to get some computational methods, machine learning and others, it makes it a lot more easier. And then also in terms of technology, being able to design things in silico before you bring it into the lab so that you can minimize the amount of waste, because in resource-limited settings, you cannot always afford all the waste and the experimentation. So getting the necessary technologies to be able to do things in silico before getting onto the lab will help in some of those areas.
All right. Thank you very much. I think we have a hand up. We probably would take that just to bring some variety to that. So Manu, please, I see your hand is up. Do you want to take this? Thank you. It's a pleasure.
Sorry. Also writing on chat, but I'll take this opportunity. Fantastic talks. I was wondering if both speakers, and maybe much more for you, Till, could speak on the regulatory side of this, and especially, in the context of India specifically, are both of the tests, either of you had to go through local regulatory processes, and where are you in that space? And this is a longer discussion, hopefully, that we should all plan for a discussion meeting, but I'm just curious on both the ends, what did that look like currently?
Yeah, so yes, you need to go through a regulatory process, and in our case, we go through. It depends a bit. So what you want to do. So there is the challenge which you have is if you come with a new diagnostic, your regulators need to understand what you are trying to do there, and so sometimes you're put into a strange pot and compared to tests which are not what you had intended to, and the same happened to us. So we are currently we have had interaction with CDESCO, so the national regulator in India. We are able to do our tests, but we don't have regulatory approval. So what we currently do is we do the testing under the conditions and of the intended use. Now, this is one first step down the path of regulatory approval. So we need to set up manufacturing and all these aspects. So that is an aspect which needs to still to come. So if you want, we are at an early stage for that, but we understand the pathway for this. Regulatory approval then comes also through the state governments, and this is another thing which we are addressed through the situation in India. As you go further, of course, every time you go into a new area of responsibility of your regulatory authority, then you need to again address this, and this is something which all of us is a challenge for all of us, as I mentioned before.
All right. One more specific question to you, Till, on how do you manage the trade-offs between limited specificity of the dipstick test as well as the relative use of ACE together with respect to some of what you are doing specific to India?
Yes. So a very good question. So here we need to understand or be aware of what we are doing with this test. The test is not there to make a decision on taking antibiotics or not. This is a test to enhance the understanding of the individual in the community, which is otherwise not seeking medical care, that they have a urinary tract infection and they should seek medical care. And that leads then to them if it's positive, those individuals will be they're entering the healthcare system and then approaching standard care. So the decision of giving an antibiotic or not is based, of course, on clinical signs and symptoms. In the future, the dipstick test may be considered as an added information, but it's predominantly the doctor who makes that decision. So it's not to make treatment decisions in the community. It's about enhancing the willingness and giving people the means to access medical care. That's the aim. So that's the trade-off, if you want.
All right. Thank you very much. I do want to apologize that we may take 5 minutes of your time. We are actually about the hour to ending this meeting, if you do not mind, because I see some really interesting questions here that I just want the panelists to take turns to answer them, if you don't mind. So 5 minutes of your time would be very much appreciated. And that takes me to the next question that generally will talk about what we want to will be what will frugal diagnostic look like in the next decade or so? What are some of the priority areas we should be looking at? I think the presentation today, we had Till speaking largely about AMR, and we also saw what Prof. Duedu had done with COVID-19. So the question is, apart from AMR, what are some of the areas we should be looking at in terms of needs for point of care that we could use frugal diagnostics to address? We can have Prof. Kwabena take 10 on this question, and we hear from Till.
All right, thank you very much. I think that when it comes to frugal diagnostics, what we could possibly be looking at is how to develop the tests that are easily implementable within our respective settings. So one of the things that we've realized is that usually there are a few equipment sitting here and there. And if we could look at doing, for example, a survey to look at which equipment for diagnostics are available and what capabilities are there, we can tailor the activities around that. Usually the danger is that we develop tests and technologies without taking into consideration the capabilities that are already existing in the places where they will be used. And so I think this is something that frugal diagnostics can look at. Keep a hand up on knowing exactly what diagnostic capabilities are at the different areas. And then at every point in time, we can tailor technologies around those ones. Thank you.
All right, final response from Till to that question, and then we have a closing remark.
I would say go and listen to and speak to the users in terms of where the biggest needs are. So when we went out with UTI in India, and then when we went to primary healthcare centers, they said, "Oh, why are you working on UTI? Why are you not working on respiratory tract infections?" Of course, respiratory tract infections, big issue, and diagnostics would be very useful for that. Why are you not working on TB? TB is a big issue. Why don't we work on diagnostics for that? We were asked. It's not just TB, it's TB and others. And clearly there are needs by numbers. So where are the biggest needs? Sexually transmitted infections, big needs. But then there are also the non-communicable diseases where you may be able to provide insights with the diagnostic at point of care. I would really echo Kwabena there. So it's the look at the capabilities, the capacities in these respective settings you're addressing. Look at the gaps and then start developing there. We have made good experience with engaging with the innovators from those respective settings, engage them, bring them, and this is what this network does, bring them into the mix of discussion, connect them with other people who have potential technology solutions but are not aware of the actual needs in detail. So if you bring those together, you can come up with good solutions. And don't focus only on technologies. Bring other aspects in. Bring behavioral sciences. Bring in aspects of drug development, etc. So it's really always a health systems approach one should take.
Thank you very much, and we hear final words from Mywen.
Thank you very much, Harry. So my name is Mywen Kersaudy-Kerhoas. I'm a microfluidic engineer in Hertford University in Edinburgh, Scotland. I'd like to thank very much Harry and Elena for supporting the delivery today of the second meet and greet meeting of the Global Frugal Diagnostic Network. Thank you to our wonderful speakers for the really inspiring talks and inspiring so much of a lively discussion, which Manu Prakash suggests we take to Slack. And I'll have a word about Slack. I don't want to take too much more of your time. I just want to put a few things on your agenda. So first of all, our third meet and greet meeting will be in October. Tentative date is 11th. We'll try to confirm that as soon as possible, and we'd really welcome your suggestions on what you want to discuss at the next meeting. Then in December, we'll have an in-person workshop in Accra in Ghana, and we'll put these effort towards writing a manifesto for publication, getting started on our sort of SOPs and our guides for the development of diagnostics in LMICs in particular. We'll also work on activity templates, and we'll kickstart some clinical and engineering collaboration around clinical needs. Next slide, please. So other activities that might be of interest to you, there's the ReClone Symposium in October. There's the Biomedical and Biotechnology Conference in Kenya in also end of October, beginning of November. Bioengineering for global health, especially those of you who are in the US, mid-November. And the African Society of Laboratory Medicine annual conference is mid-December in Cape Town, so right after our December workshop in Accra in Ghana. Finally, quick words on communication. It's really important. Please do get the word out about the Global Frugal Diagnostic Network. Let's get the community involved, and let's listen to the community on what is really needed on the ground. We have a LinkedIn account. Please participate and engage in these. And we have also the Twitter account. Please, retweet or ask anything on Twitter. We've also started a Nature Microbiology blog series. So I've posted earlier in this discussion 2 links to the first 2 of these blog posts. So it's a good way if you want to know what the network is about and hear what we've been doing recently. We have a Slack as discussed. It's very much in development. It's a little bit quiet at the moment, but we intend to make full use of the technology and get the activity starting. One of our ideas was to have the paper of the week on Friday. People post a paper and say why they like it, etc. And we hope that could be, a fun activity for everybody to engage in. And finally, we have a website which is in construction, and we hope that before December it will be live. So now I'd like to thank all of you for joining this meeting. I hope you've enjoyed, and I hope we will hear from you again very soon. Thank you so much. Have a lovely day or a lovely evening, depending where you are.
Thank you very much.
Thank you all for coming.
Thank you all for coming. Bye.
Thank you very much. Bye-bye.
Bye. Thank you
Bye.
too.
Bye.